Hey, thank you, Kate, for that intro, and thank you everyone for coming. Just going to give you an overview of what I've been up to the past 5 years. And so as the title of my project implies, my work focuses on plant responses to projected changes in our climate, particularly higher air temperatures, drought, and elevated CO2. So I'd like to start off briefly discussing some of these projected changes. So I'm sure that all of you have seen some version of this figure that depicts the global projections of atmospheric CO2 concentrations based on various emission scenarios. And here you can see that if we don't curb our fossil fuel emissions, we can expect as high as a threefold increase in atmospheric CO2 by the end of the century. And so as CO2 and other greenhouse gases have been increasing, our temperatures have also been increasing. And so these figures right here are showing the average temperature increase predicted for the future based on a high emission scenario and a low emission scenario. And what's important about these two figures is that for one, they show that even under the lowest emission scenario, we're still expecting to see an increase in temperature. And two, they show that the greatest change in global temperatures are going to occur at the northern latitudes. And so there's no doubt that increasing greenhouse gases have contributed and will continue to contribute to rising global temperatures, but much of the projected change is due to an increase of heat wave events. So these figures here are showing an increase in the severity, duration, and frequency of heat waves by the end of the century. And so this first figure, you can see. Alright, so this first figure is showing that the average high temperature of a heat wave is projected to increase. The average duration of a typical heat wave is going to increase, and then the number of heat wave events per year is projected to increase. And so here, this is a thermal image over Greenland that was taken in 2016 during a heat wave event. Here is Greenland, here's the US, Canada. And so what's important here is that it's showing that heat waves are occurring everywhere across the globe, but their effects may be more severe in the boreal forest because plants there aren't used to experiencing such high temperatures. And so in this particular example, you can see many regions across Canada where the highs got above 30 degrees. And in fact, just last week, parts of Alaska experienced, or they set their all-time high record. And so here, this is from Anchorage, and this was on the 4th of July. It reached 90 degrees. Okay. So in addition to our futures being warmer, they're also going to be drier. And so this figure is showing the prevalence of drought by the end of the century. And here you can see that the warm colors are showing areas where drought is expected to increase. And you can see that those warm colors make up the majority of the figure. So we know that heat and drought events are expected to increase, but let's take a look back and see what damage these events have already caused. And so this image is showing locations where past extreme heat and drought events have led to forced mortality, and those are indicated by the dots or the oval shapes. And so this is showing, so this figure shows drought and heat-induced mortality are already being observed across all major forest biomes. So it's clear that many species are being negatively affected by these extreme heats and these extreme drought events. But now I'd like to focus on specifically two of them. Those two are white birch and, paper birch and white spruce. And they have been declining in recent decades as our temperatures have been warming. And it's been suggested that the availability of climate suitable habitat for these two species is presently shrinking. Okay, so paper birch, which is shown in this image right here, it's a shade intolerant, fast growing angiosperm that lives for typically around 80 to 100 years, but in stressful environments it can live for fewer than 40 years. In contrast to this species, white spruce, which is shown down here, is a very slow growing conifer that can live upwards of 200 to 300 years. And both these species are predominantly found in the boreal forests, so their ranges overlap, where they're typically found in the northern part of the U.S., in the upper Midwest, all across Canada, and then into Alaska. And so these specific reasons why I chose to study paper birch, well, there are a few of them. The first was that these are two species that are declining here in the state of Wisconsin. And birch, and white spruce specifically has seen a decline, has seen an increase in mortality by 85% since 1996. So that's a huge increase. And paper birch, actually within the state, their annual net growth has been negative since 2009, which means that their mortality now exceeds their growth. Secondly, both of these species are important for the forest products industry, particularly spruce has their timber is very versatile. And lastly, we know relatively little about how these two species respond to heat, particularly heat wave stress. And so the overall goal of this project was to examine species-specific responses to the combination of heat wave and drought stress under elevated CO2 to determine their ability to acclimate to future climate change type scenarios. And so we did this by conducting a multiple organizational level analysis that examined physiological responses, hydraulic responses, and biochemical responses. And by understanding whole plant multi-level responses to stress, it will improve our ability to predict how these species will respond in the future. Sorry, I'm having trouble with the mouse, yeah. It's not there.
Oh, it's because it's actually, it's actually not showing up. So you need your notes, there we go. It's back.
Thank you. All right, sorry, where was I now? How do plants respond to stress, such as heat and drought? Well, there are many different types of response, and I'm sure you're all well aware of that. But I chose to focus on the physiological, biochemical, and hydraulic responses. So here are a few common responses of each of these types. So a common physiological response to stress has to do with altering gas exchange, and so there may be changes in stomatal reductance or photosynthesis. Plants can also alter their metabolite composition when they're stressed, so they may accumulate osmolytes or antioxidants, and that would be a biochemical response. And they can also make adjustments to other traits that affect how water is transported through a plant, such as modifying xylem anatomy or changing water potentials. So those would be considered hydraulic responses. Okay, so here's a brief outline of the following sections. I'm first going to go over the experimental design And I'll talk about the physiological responses, the biochemical responses, and lastly, the hydraulic responses. So for this project, we had one main experiment, and it took place over two consecutive years. It began in 2016. We started with Wisconsin native paper birch and white spruce seedlings, and once they established, we moved them into the Biotron facility where they grew for the duration of the 2016 and 2017 growing seasons. So within the Biotron, the greenhouse rooms, all the greenhouse rooms had their CO2 concentration elevated. The daily temperature within these rooms, they were identical except for periods when heat waves occurred. And that, in heat waves, and half the plants experienced heat waves, which lasted for 8 continuous days, where the temperatures were increased by 10 degrees. And so these heat wave events happened in June, July, and August of both years. And so this figure is showing the temperature in 2017 of just 2 of the rooms. So the black would be a room that never experienced a heat wave, and the red is obviously a room that did experience the heat wave. And so you can see that the temperatures are identical except for those heat wave periods. So we also manipulated the watering machine as well. So half the plants in each room were well watered, the other half were reduced watered. And so we have 4 treatment groups that I'm going to be referring to when I describe the results. And so the controls, these plants were grown under elevated CO2. They were well watered and they never experienced heat waves. The droughted plants were grown under elevated CO2. They were droughted and they never experienced heat waves. The heat wave treated plants, they were well watered, grown under elevated CO2, experienced heat waves. And then lastly, the combined stress, the heat wave and droughted plants were heat wave, droughted, and then grown under elevated CO2. And this is the color scheme that I use on my figures too. So, okay. All right, so this finally brings us to the first part of the project where we ask the general question: how does heat, drought, and elevated CO2 affect gas exchange, respiration, and growth differently in birch and spruce? And so I'd first like to take a second to discuss some of the typical physiological responses plants have in a little bit more detail. And so when plants experience drought, that's a condition when they experience low soil water availability, and that low soil water availability is often accompanied with increasing vapor pressure deficit, which is a metric that indicates to us how the plants are feeling the evaporative demand. And so during a drought condition, VPD is going to be high, and so water is going to be pulled out of the leaf. And so if water is being lost from the leaf, it has to be replaced by the plant's hydraulic network. But if it's in a drought, there really isn't any water for it to replace. And so the plant risks desiccation. And now, to prevent that, what they typically do, the typical first response is to close their stomata. And so by closing their stomata, they prevent water vapor from leaving the plant, and by doing so, they conserve water loss. But there's a consequence of closing their stomata, that is that CO2 can no longer enter. And so when plants experience drought, they close their stomata to conserve water loss, but by doing so, they also reduce photosynthesis. Now, in contrast to drought stress, the first response plants have to heat is not typically water conservation, and this is because as air temperatures increase, so don't the leaf temperatures. And so as a means to cool those leaves and prevent them from exceeding their thermal limits, plants open their stomata and they transpire more. And so by transpiring, they can reduce their leaf temperatures and prevent damage to their photosynthetic machinery. And so now, what happens when both of these stresses still occur? Well, a conflict, right? This conflict arises within their stomatal response. So either they're going to use water, open their stomata, keep their leaves cool, but then risk desiccation, or they're going to close their stomata, but to save or to conserve water, but then risk causing damage to their photosynthetic components. And so, so far, species responses to the combination of high temperatures and drought have been pretty mixed. So there's no consistent response across species. And so here's another environmental factor that plants are going to have to deal with in the future, and that's elevated CO2. And plant responses to elevated CO2, these have been well studied. So when plants experience high CO2, they typically respond as they would to drought. They reduce the monoconductance. But in doing so, photosynthesis is usually maintained or even increases because of all that extra available carbon around. And so high CO2 causes monoconductance to be reduced, but photosynthesis is often elevated. And there's been some previous work that has shown when elevated CO2 is combined with drought stress, or when it's combined with heat, it can mitigate the effects of heat and drought. But we really don't have a very clear understanding of how plants respond when all three factors, when they're experiencing all three of those factors. Okay, so our first hypothesis is that we predict that heat wave and drought treatment will reduce growth and gas exchange, but these reductions will be greatest when both stressors co-occur. Secondly, we predict the effects of combined heat and drought stress will be more severe in spruce based on observed declines in natural populations. And lastly, given the recent declines of both of these species, we predict that thermal acclimation of photosynthesis and respiration will not occur after 2 years of heat wave stress. All right, and so we quantified photosynthetic acclimation 2 ways by 2 methods that are based off of a typical temperature response curve. And so the temperature response curve shows temperature on the x and photosynthetic rate on the y. And so photosynthesis increases to some point, but then declines as temperature further increases. And so the temperature at the peak of the curve is considered the temperature optimum, and then the photosynthetic rate at that temperature optimum is the photosynthetic optimum. And so for acclimation to occur in warm plants, those plants would show a shift in their temperature optimum. So the temperature optimum would go from a lower temperature to a higher temperature. But what's important about this is that the photosynthetic optimum does not decline. So for acclimation of the first type, it involves a shift in the temperature optimum, but as long as the photosynthetic optimum doesn't decline. So, the second type of acclimation does not involve a shift in the T-off, but it actually it involves the shape of the curve becoming wider. And so if the gray curve is a not warm plant, the black curve would be a warm plant. And so by having a wider curve, it allows for a higher photosynthetic rate across a greater range of temperatures. And so these are the 2 types of photosynthetic acclimation that we're looking for. Okay, so this next figure is showing an example of what respiratory acclimation would look like. So the red bars are plants that were grown under elevated temperatures, the white bars are just ambient grown. And so what you'd expect is at an elevated temperature, the plants that were grown at that higher temperature would have a lower rate of respiration. And so if I were to overlay plant respiration in response to temperature, respiration would continue to increase well past the point where photosynthesis declines. And so this is important because if photosynthesis acclimates but respiration doesn't, then the ratio of carbon assimilated to carbon consumed decreases and overall growth decreases. And so this is why it's important that both processes acclimate. All right, so to address our hypotheses, we measured gas exchange multiple times over both growing seasons. we were most interested in net photosynthesis, which is gross photosynthesis minus respiration, and we were also interested in stomatoconductance, which is the rate of water vapor leaving stomata. We also generated temperature response curves at the end of the first growing season, at the start of the second growing season, after the first heat wave of the second season, and then at the very end of the second season. And we all then we measured acclimation of respiration, which was measured at lower temperatures and then at elevated temperatures. And lastly, we measured stem diameters at the start and at the end of each season, and from those we were able to calculate basal area growth. All right, so this first figure is data from Birch. The top figure is showing the photosynthetic rate, the bottom figure is showing stomatal conductance. The first half of the figure, this is data from 2016, this is from 2017, and then the areas that are shaded indicate when heat waves occurred. And so what I want you guys to focus on here are the points that I have highlighted. And so what it's showing, so the X's are the plants that receive heat and that receive the heat and drought treatment. So what it's showing is when a heat wave occurs, there's a decline in photosynthesis, but then afterwards they recover. There's a decline and then they recover. And so this pattern can be seen both in photosynthesis and in stomatoconductance, and it's consistent across both years. And so to take all this information and try to boil it down, we looked at the seasonal averages. And so what we found at the end of the first year, the plants that were droughted reduced their photosynthetic rates. The plants that were heat and drought stressed, they reduced their photosynthetic rates. But in the second year, these plants somewhat recovered, whereas the plants that received both stresses did not recover. Similar pattern can be found in stomatoconductance, where the first year of stress caused a reduction in all treatment groups, but the individual drought and the individual heat treatment, they recovered by year 2, where the plants that received the combined treatment did not recover. So spruce shows a very different response. They don't seem to be as affected by those heat wave events. And so if you look, again, at focusing on the dots, a heat wave occurs, there's really not much of a change. Last heat wave, not much of a change, except for this decline right here. Generally, there's not a major decrease in either photosynthesis or stomatoconductance in response to heat waves. And in 2017, you can see that all treatments are fairly consistent. And so, moving on to look at the seasonal averages, in the first year, we found that, like the birch, drought reduced photosynthetic rates, and those reductions were most severe when drought occurred with the heat waves. But by the second year, all treatments recovered. The same can be found in stomatoconductance. The first year, the heat and drought plants severely reduced conductance, but by year 2, everybody recovered. All right, so these next 2 figures are showing the photosynthetic acclimation parameters that we were interested in. The first one is the temperature optimum. The second is the photosynthetic optimum. And so again, these are data from the end of the first year, the start of the second year, after the first heat wave, and then at the very end of the year. And regardless of date, you can see that that the temperature optimum never differs among any of the treatment groups. So then we went to look for the next type of acclimation, which was a shift in their temperature response curve. And so this was from the end of 2016, this is from the end of 2017, and you can see that these curves are nearly identical. So we found no evidence of photosynthetic acclimation in the birch. The spruce responded somewhat differently, where 1 year of heat wave stress, we found no differences. At the start of the next year, we found no differences. But when the plants experienced heat for the first time, they increased their temperature optimum compared to the plants that hadn't experienced that heat. However, by the end of the year, we found that the non-heated plants eventually increased their temperature optimum, and we found no differences between the treatments. And we found no differences in the photosynthetic optimum. And so this is an example from this point, and it's just showing that we found no shift in the curve. And so to us, what does this indicate? Did they acclimate or did they not? And so it shows that spruce adjusts their temperature optimum seasonally. So they start off at a lower temperature earlier in the year, and then they increase it to the end of the year. And by having exposure to elevated temperatures at some point during that growing season, that led to an earlier adjustment to the seasonal max. But they never exceeded that seasonal max. So it looks like that they do have the ability to photosynthetically acclimate, although it may be minor. Okay, going back to this figure, again, so for acclimation of respiration, you'd expect plants that were heated to have a lower rate of respiration at an elevated temperature. And so here, this is for the Birch data, and that's exactly what we found. The plants that experienced heat drought stress did reduce respiration in comparison to the control plants. And this was not true for spruce. All right, and lastly, we looked at basal area growth from 2 years of just treatments, and so the first figure is for Birch, the second is for spruce. The gray symbols are from data from 2016, and the black are from 2017. And overall, what I want you to look at is the points that I have circled. And so it really drives home the point that 2 years of heat and drought stress did lead to significant declines in plant growth. And so just quickly to summarize these findings, in Birch we found that gas exchange and growth declined. There was no evidence of photosynthetic acclimation, but there was some evidence of respiratory acclimation. In the spruce, gas exchange was shown to recover in year 2, and these plants did show some minor evidence of photosynthetic acclimation, but not respiratory acclimation. And so, moving on to whether or not our hypotheses were supported, we found that our first hypothesis was supported because we found that the individual heat and drought treatments only had minor effects. Whereas when the 2 stresses combined, they really did have major consequences on gas exchange and growth. Second, we found that Birch was actually more adversely affected by the combined treatment, which was contrary to what we expected. And finally, the data show that both species have a minimal capacity for thermal acclimation because we found that both processes were not adjusted, and the adjustment in respiration in the Birch, this was most likely due to a carbon imbalance, because if you remember, they had really, really low photosynthetic rates at that time as well. And even though we saw the heat wave treated spruce, they did recover gas exchange, and they did show some evidence of photosynthetic acclimation. Those minor adjustments that they made weren't enough to prevent these species from showing growth declines. All right, so now for the next part of the project, we asked the general question: are biochemical responses to heat and drought stress distinct from the responses to either independent stress? And so when I say biochemical responses, I'm actually only referring to changes in metabolite content. And so plants, like any living organism, they're constantly making adjustments to maintain cellular homeostasis, which is a condition of internal equilibrium or stability. And so as challenging as that may be on a day-to-day basis, it's even more challenging when environmental conditions sharply change, as they do during a heat wave. And so when plants experience, say, high temperatures, drought stress, for example, their internal environment is significantly disrupted. But there are a few, well, actually there are several ways that they can restore their cellular homeostasis, but I'm just going to describe a few of them. And so one way is to increase osmolalite content, so these may be compounds such as proline or other amino acids or possibly sugars. And the role of these compounds is to maintain target pressure, which allows the plant to remain functional. They can also adjust the ratios of various ions, which are also important for maintaining cellular function. And the last example I wanted to mention is a change in antioxidant production. And so plants can increase their antioxidants, which help combat oxidative stress, and then which can then lead to a restoration of oxidative homeostasis. And so these examples are common. So plants will respond this way under multiple stresses. It's not so. What I just described is not unique to one type of stress. And so you might expect, though, if a plant experiences multiple stresses at once, that they may enhance the accumulation of these compounds. But so far, species responses to heat and drought stress have been mixed, where some compounds have been found to accumulate only when multiple stresses occur, but not when the individual stresses occur, whereas others have shown, regardless of whether it's a combined stress or the individual stress, the metabolite response is still the same. And but most of these examples that I'm discussing right now has come from work that has been on herbaceous species, and so the uncertainties around these responses are even greater for woody species. So briefly, I just want to talk about two important antioxidants that have been shown to increase plant thermal tolerance, and they are the tocopherols and carotenoids. Tocopherols are compounds which are embedded within the thylakoid membrane, and they prevent lipid peroxidation. Carotenoids are pigments that are often associated with photosystem protection. And so when plants experience stress, such as one of these stresses, there's usually an increase in the production of reactive oxygen species, which are commonly radicals. And those radicals lead to oxidative damage, but plants produce antioxidants in order to combat the activity of those compounds. And so, right here, this figure is showing alpha tocopherol, which is vitamin E within the thylakoid membrane, and here you can see that it's neutralizing these radicals that otherwise would have caused damage to the phospholipids. And so by having more of these protective compounds within the thylakoid membrane and around your photosystems during stress, they can help maintain photosynthetic activity, thereby improving plant thermal tolerance. And studies have shown that plants that accumulate more of these compounds do have increased thermal tolerance. Okay, so moving on to our hypotheses. First, we hypothesized that the heat and drought stress will result in a unique accumulation of metabolites. Two, given the differences observed in physiological responses between the two species, we also predict that the biochemical responses are going to be species specific. And lastly, due to the recent decline in birch and spruce, we expect that antioxidant content will not be improved or will not increase under heat wave stress, and therefore it won't lead to an increase in plant thermal tolerance. So to address these three hypotheses, we measured a variety of stress-related primary and secondary metabolites, but today I'm just going to talk about some of the few interesting results. And so we collected leaf tissue samples at the start and at the end of each growing season, and then analyzed them for free amino acids, carotenoids, and tocopherols via spectrophotometry or high-performance liquid chromatography. We also measured leaf thermal tolerance, and the way that this was done is we collected leaf samples and then subjected them to temperatures from 25 to 60 degrees. And then after they were exposed to those temperatures, we measured photosynthetic efficiency, which was done through chlorophyll fluorescence. And so from those measurements, you can generate a curve that shows photosynthetic efficiency and temperature. And so the metric of interest to us is the T50, which is the temperature that causes a 50% decline in photosynthetic efficiency. And so plants that have increased thermal tolerance, you'd expect a shift in the T50 to a higher temperature. Okay. So of the 20 common sodium-genic amino acids, we were able to quantify 18 of them. In addition, we quantified GABA and ornithine. I am not going to go through all of these, so don't worry. I just want to point out which amino acids responded uniquely to heat and drought stress. And so this is for Birch, and here you can see that we only found 2 unique responses, and these occurred in tryptophan and phenylalanine, and they were both in the first year of heat stress. And the significance of the increase in these 2 amino acids is that they are precursors to a wide variety of secondary metabolites, and many of these compounds that are derived from these 2 have significant roles in protection against abiotic and biotic stress. And so I don't expect you guys to be able to read this, but I just want to put this up here to show you that this is phenylalanine, and these are all these different compounds that can be derived from phenylalanine. And so the accumulation of these 2 compounds to us says that, yeah, okay, so these 2 increased, but that must mean, based on everything else that's derived from, that there had to have been other unique responses. But so in addition to only finding 2 unique responses in Birch, that was surprising, but I think what was most surprising was the lack of response in proline and GABA. And these are 2 osmolytes that have been well studied. They're known to increase under osmotic stress, and yet we found no changes. And so perhaps some of the compounds that were synthesized from tryptophan or phenylalanine may be providing some sort of osmotic protection to replace proline and GABA. But anyways, that's pretty interesting. Okay, moving on to spruce. All right, so this is where things really get kind of crazy, because we found that the birch plants, the spruce that experienced heat and drought did have several unique responses. And so again, I don't have time to go through all of these, but I'm just going to focus on a few. And so, similar to the birch, we showed an increase in tryptophan and phenylalanine in the first year of heat stress. So didn't the spruce. We also found proline and GABA. This was upwards of a 10-fold increase in proline when the 2 stresses co-occurred. That didn't occur when only when plants experienced drought. And so what was also unexpected was we saw all these different changes in amino acid content in year 1, but then when we looked at data from year 2, we found no differences. And so this is saying that that these plants must have made some major changes in between year 1 and year 2. Okay, so next we looked at the antioxidant content to determine if it increased under heat wave stress, and if it did that lead to an increase in thermal tolerance? And so this first figure is of carotenoids for Birch, and as you can see, at each of the 4 sampling points, we found no differences. Next, we looked at alpha tocopherols. Again, this is vitamin E. This is the one that's found in the thylakoid membrane. We found that it tends to increase with plant age, but we found no changes in response to heat wave stress. So when we looked at the thermal tolerance curves, it wasn't surprising that we found that these plants did not, plants that experienced heat wave stress did not improve their thermal tolerance. Spruce also didn't increase their carotenoid content under heat waves, but they did increase alpha tocopherol, not in year 1 where we saw those crazy changes, but in year 2. Both the heat wave and the heat plus drought increased vitamin E. And so, that led to, or most likely led to, an increase in thermal tolerance. So here the control, their T50 compared to the T50 when both stresses co-occurred. And we showed that there was a positive relationship between the alpha tocopherol content and the T50. Okay, so to summarize this, we found few unique responses in the birch. We found no changes in antioxidant content in those plants either, and we also found no change in thermal tolerance. In the spruce, we found major changes in year 1. They did increase vitamin E in year 2, and we did find that thermal tolerance was improved. So, so the data for spruce does support our first hypothesis, but it doesn't for birch, where the spruce showed that the combined stress did result in many unique responses from either independent stress, and so, but these responses again were only limited to year 1, which suggests that they had restored cellular homeostasis by the 2nd year. In contrast to the spruce, the birch showed only a minor response in the metabolites that we measured. Secondly, we found that the two species responded very differently to heat and drought, which was similar to the physiological responses that we measured. So this is evidence for our second hypothesis. And lastly, our third hypothesis was supported by the birch data, because we thought that because these plants were already declining in nature, that they must somehow lack the ability to produce compounds that would make them survive in these stressful conditions. And so we found that was true for the birch. They didn't adjust their antioxidant content and they didn't improve thermal tolerance. But the spruce actually surprised us. They increased alpha tocopherol and they increased thermal tolerance. And so these findings suggest that white spruce may be more capable of adjustment under multiple stressors, whereas the birch may not be, and that their lack of ability to metabolically adjust may increase the likelihood that these plants are going to experience greater growth declines in the future. All right, so moving on to the third and final part of the project, which focuses on plant hydraulic traits. We asked the general question, how do plant hydraulic traits respond to the combination of heat waves, drought, and elevated CO2 and species with contrasting wood types? And so this image right here is a cross-section of birch, and here I'm pointing to the vessels. This is how this species typically moves water throughout its stem. Down here is a cross-section of a spruce stem, and here you can see all these tiny tracheids, and this is how that species moves water throughout its stem. All right, so plant hydraulic traits are traits that are related somehow to the transport of water within a plant. And so hydraulic responses are typically tightly linked with the plant's water status. And so these hydraulic responses to osmotic stress have been heavily studied in the past. We have a pretty good idea of how plants respond to drought stress, but much less is known about how these hydraulic traits are altered when drought stress occurs with heat stress and elevated CO2. And so when plants experience drought, they can make immediate adjustments that will help them, that will improve their chances of surviving through that drought stress. And some of these responses I've already discussed in previous sections, but because they are somehow tied to water transport, they can also be considered hydraulic traits. And so here I'm showing that under drought stress, plants reduce stomatal conductance, reduce stomatal conductance, they reduce, they restrict the loss of water to the atmosphere so they conserve water. So that's usually an immediate response. And the other example I provide is that they can increase their solute concentration. And so by doing that, that increases turgor pressure, which allows for cellular function to continue as water stress may increase. And so here you can see a flaccid cell, the plant is wilted, it's not happy. Here's a turgid cell, the plant seems pretty happy. So in addition to these immediate adjustments they can make, they can also modify permanent structures that will help them better tolerate osmotic stress in the long term. And some of these changes can be made to new wood or to new leaves. So for example, they can alter their conduit dimensions where they can make their conduits narrower, and they can also build their cell walls to become thicker. And so these structural modifications reinforce the conduits against collapse. And so if you think about a plant being under drought stress and evaporative demand is high, so you can picture the pull of water through the plant being very strong. And so if the conduits have thick cell walls and their areas are smaller, the likelihood that they're going to collapse under that pressure is low. So in addition to those structural changes to the conduits, they can also produce leaves that are smaller, they can produce fewer leaves, they can adjust the stomatal properties on those leaves. And these are just a few of the structural changes that plants can make. And so for this part of the project, we hypothesized that because the physiological and biochemical responses were species-specific, we also predict that the hydraulic responses are going to be species-specific to heat and drought stress. Second, we predict that hydraulic adjustment will be greater in birch because their wood type is generally more vulnerable to water stress. And third, we predict structural modifications will be made to the leaves and conduits in the second year to help them tolerate osmotic stress. And so for this section, we actually measured several functional and hydraulic traits, but I'm just going to focus on a few of the structural traits. And so we measured leaf areas at the start of the second year. We looked at stomatal density at the start of the second year. And so here's an image of a leaf surface from birch, and these are the stomata. This image down here is showing a spruce needle, and you can see the stomata in 2 rows. And also we measured conduit resistance to collapse. And so to do that, we measured the thickness of the cell wall to its span. And like I previously mentioned, the more reinforced the conduit is, the better it's going to withstand under increasing water stress. Okay, so these 2 figures are for the leaf traits in birch, and what we found was that in the second year, the birch produced leaves that were much smaller, but we found that we found no differences in stomatal conductance. And so by producing smaller leaves the second year, this indicates that the plants may have been adjusting their water use strategies and potentially trying to reduce how much water they lose by constructing leaves that are much smaller. Next, we looked at the structure of the conduits, and so the scale is showing increasing resistance to conduit collapse in 2016 and 2017. And I want you just to focus on the point I have highlighted here. And this shows that birch did produce conduits that were more structurally reinforced the second year, so after experiencing heat and drought stress, where the other treatments really made no adjustments. Spruce shows a very different response. So this first image is leaf area, where we found no differences here, and then still model density. We found that under the combined treatment, it didn't differ from the controls. And then we went to look at conduit resistance to collapse. And so this first figure is showing data from the early wood. So early wood is produced at the first part of the year, and we found by year 2, no differences. And then down here, this is showing late wood. So this is wood that's put on later in the season. And we found that the heat wave treatments really had no effect on increasing conduit resistance to collapse. The only treatment that had an effect on that was the drought-only treatment. All right, so along with the previous, with the findings from the previous sections, hydraulic responses to the combined heat and drought stress were species-specific. Birch made some modifications to their structure, whereas the spruce did not, and his lack of adjustment in spruce could be due to their overall growth strategy, where it's a slow-growing species, it's long-lived, so it's possible that in order to ensure its survival, that it had already constructed needles and conduits that were able to withstand osmotic stress. Second, we found adjustment of these traits was greater in birch, so this supports our second hypothesis. And lastly, we found that structural traits, changes to structural traits were only made in birch and not in spruce. And so, okay, quickly I'm going to go over my final conclusions here. So yeah, so to wrap everything up, we showed physiological, biochemical, and hydraulic responses to 2 years of heat wave stress differed significantly between birch and spruce. The birch reduced gas exchange and growth. They had a minimal capacity for thermal acclimation and biochemical adjustment, but they did seem to show some modifications to their structural traits. But overall, they showed a general lack in adjustment from year 1 to year 2, meaning that those negative responses that we saw in year 1, they didn't improve by year 2. In spruce, we found that they had reductions in gas exchange that coincided with elevated metabolite content, but this only occurred in the first year, so those responses disappeared by year 2, which may point to stress memory playing a role, where the first year of stress may have primed their stress response systems for a more prepared response the following year. Second, we showed responses to combined heat and drought stress were often unique under the combined stress compared to those independent stresses, which supports some previous work that has shown high temperatures experienced during a prolonged drought do have major consequences, particularly on carbon metabolism. 3, our findings highlight the importance of a multi-level analysis. So, for example, if we were to focus on the anatomical traits if we were to focus strictly on the anatomical traits, we would have led to believe that, yeah, birch is, they are adjusting, they may be doing okay in the future, but when we take all of these results together, so the biochemical, the physiological, and the hydraulic, you can see that these plants really aren't making the adjustments that are necessary to survive, and they are actually performing pretty poorly under heat and drought. Fourth, our findings also highlight the importance of long-term experiments when studying plant responses to stress. This is because not all responses emerge within that first year. And so if we were to base our predictions of how spruce was going to respond in the future based on their first year responses, we may have been led to believe that they are not going to do well in the future because we saw major declines in gas exchange and growth, and they kind of freaked out with their metabolites. But by year 2, gas exchange recovers. They appear to restore homeostasis. They appeared to restore cellular homeostasis, and so that tells a different story. And so Lastly, based on this 2-year experiment, our predictions for the future are predictions for spruce in the future are optimistic because they have shown the ability to thermally acclimate to future environmental conditions, although their growth may be slowed, which may put them at a competitive disadvantage. In contrast, the future for birch appears somewhat grim. Their inability to acclimate to high temperatures and drought stress may restrict their future distributions to only narrow pockets of wet and cool habitats. And so with that, I'd first like to thank my advisor, Kate, for allowing me to take on such a diverse project and really never saying no along the way. Also, I want to thank her for how much time she's put into to editing my drafts. It's probably days, weeks at this point, so I really, really appreciate that. And for generally just keeping me on track. I'd also like to thank my committee, Ken, Eric, Brett, and Roshi, for their guidance along the way. In general, I'd like to thank the McCullough Lab. Also, Rakesh and staff from the Forest Service, Bjorn from the Biotron, Jesse and Ben from the Isotope Lab, and then all the undergrads that helped over the summer. So Kevin, Sam, Ed, Nick, Nina, and Kylie. And thank all of you for coming, and I'll take questions.